Welcome back to The Explainer, everyone. I'm Dr. Chem, and today we're tackling one of the absolute coolest challenges in modern biotechnology. Picture the chaotic, microscopic soup inside a living cell. You've got millions of proteins, nucleic acids, lipids, and carbohydrates, all just jumbled together in this incredibly complex liquid mixture. So how in the world do you isolate just one specific molecule from that massive mess without damaging it? Well, today we're going to learn exactly how scientists achieve this feat by becoming the ultimate molecular matchmakers.
So let's just dive right into it. Think about the molecules inside living things almost like a giant bucket of mixed magnets. They stick to some very specific molecules, but ignore others entirely. Exactly the way a magnet snaps right to metal, but doesn't do a thing to wood or plastic. In the lab, we actively leverage these natural, highly specific, and reversible biological interactions to hunt down and isolate our exact target molecule from that crazy complex mixture.
All right, here's our game plan for today's explainer. We're going to cover the magic of affinity, break down the chromatography system itself, walk through how the process actually works step by step, weigh the pros and cons, and finally, wrap up with some amazing real-world applications. Okay, let's kick things off with Section 1. The magic of affinity. So, affinity chromatography. It's a highly selective separation technique, but, unlike standard methods you might be used to like gel filtration or ion exchange, which sort things out based on just physical size, charge, or polarity, this is a whole different ballgame. It separates biomolecules based entirely on their specific biological functions or individual chemical structures. It relies completely on biological matchmaking.
So what do these matches actually look like in practice? Well, think of them as highly specific molecular locks and keys. We're relying on that natural, almost magnetic attraction between an antigen and its antibody, an enzyme and its substrate, or a receptor and a ligand. We've also got lectins binding to specific carbohydrates and metal ions catching engineered his tags. But here's the real kicker. The absolute secret sauce of this whole technique. These interactions are reversible. We can trigger them to grab on and then, whenever we're ready, command them to let go.
To actually harness all this, we need the right hardware. Which brings us to Section 2, the chromatography system.
To make this molecular sorting a reality, we need a very specific lab setup. The chromatography column is basically the physical tube where the magic happens. Inside that, we've got the stationary phase, usually a solid and soluble matrix, like agarose or polyacrylamide. And firmly attached to that solid matrix is our immobilized ligand. That's our chemical bait, right? We also use a series of buffers for binding, washing, and eluting to strictly control the chemical environment. And finally, as the liquid drips out the bottom of the column, a detector and fraction collector analyze it, often using UV spectroscopy, SDS page, or enzyme assays, and physically catch our isolated target.
All right, equipment is prepped on the benchtop, so let's get into the action. Section 3, how the process works.
There are 4 main operational phases to take us from that messy biological soup to a perfectly pure sample. First, we equilibrate the column. 2, we load the sample. 3, we aggressively wash away the impurities. And 4, we elute the target molecule. Let's break these down.
Okay, looking at step 1. We start by pumping a binding buffer through our stationary phase. Now, the absolute crucial point here is that we're creating the perfect localized environment. We're adjusting the pH and the ionic strength so that those immobilized ligands, our chemical bait, are perfectly conditioned, active, and literally standing by, ready to catch their specific partners.
Next up, we introduce our complex sample. As this biological soup flows over the stationary phase, our specific target molecules actively lock onto the immobilized ligands. Bam! Just like magnets snapping together. Meanwhile, all the other proteins, the lipids, the junk, they have absolutely zero affinity for the ligand. So, during the wash phase, we pump more buffer through and aggressively rinse all those non-target molecules right out. The only thing left behind is our target, securely tethered inside the column.
And finally, we hit step 4, elution. We've caught our target, but now we actually need to collect it, which means we have to break that specific magnetic-like hold. To do this, we change up the conditions of the elution buffer. We might alter the pH, tweak the salt concentration, or even strategically introduce a competitive ligand into the flow that effectively knocks our target off the binding site. The reversible interaction breaks, and our highly purified target molecule washes right out the bottom of the column, ready to be collected.
It sounds like pure magic, right? But let's do a quick reality check. Section 4, pros, cons, and variations.
The advantages here are honestly staggering.
You're getting exceptional selectivity, sometimes hitting a several thousand-fold purification in literally one single rapid step. It gives you incredible recovery rates while preserving the delicate biological activity of your molecule. But, we got to weigh that against the practical limitations. Affinity media, it can be insanely expensive. You also have to worry about the immobilized ligands degrading or leaking off the matrix over time. And perhaps the biggest hurdle, you absolutely must have prior knowledge of your target's specific binding partner to even design the column in the first place. No partner, no purification.
Now, because biology is so incredibly diverse, researchers have come up with a whole bunch of specialized variations of this technique. Immunoaffinity is used to isolate specific antigens, while IMAC, or immobilized metal affinity chromatography, is huge for grabbing recombinant proteins tagged with histidine. We also rely heavily on protein A and G for antibody purification, lectin affinity for trapping specific carbohydrates, plus other super robust systems like diligand, glutathione, and streptavidin biotin affinity.
All right, let's bring it all home and see how this lab theory translates into massive global impact. Section 5, Real-World Applications. Let's look at some real, hard data here. This chromatogram tracks a live purification run. Check out that really distinct, sharp spike over on the right side of the graph. That peak represents the exact split second a competitive ligand was flushed into the column, forcing it to release a massive, highly pure concentration of the enzyme horseradish peroxidase, or HRP. It's literal visual proof of this molecular matchmaking working flawlessly in real time.
And the real-world applications for this level of specificity, they're absolutely profound. I mean, affinity chromatography is basically an indispensable backbone of the modern pharmaceutical industry. It's mandatory for the large-scale therapeutic manufacturing of antibodies, enzymes, and recombined proteins. Honestly, without this technology, the safe and rapid mass production of life-saving vaccines and advanced biopharmaceuticals simply wouldn't be possible. It's the absolute driving engine behind bleeding-edge biotechnology research, proteomics, biomarker discovery, and clinical diagnostics.
So, to wrap up this explainer, I want to leave you with a thought. How might mastering this ultimate molecular matchmaking unlock the very next generation of therapeutic cures? Because, as our understanding of these biological locks and keys gets deeper, our ability to isolate and manufacture the complex molecules we need to fight diseases is only going to skyrocket. What you should really take away from today is that affinity chromatography isn't just some niche lab trick. It's genuinely one of the most powerful foundational pillars of modern medicine. Thanks for being such focused learners today, and I'll catch you in the next explainer.